Hello, welcome to this guided presentation. My name is Silvia Vanilla and I work as a scientist at MIMETAS. And today we're going to go through a poster titled A Novel High Throughput Platform for Head and Neck Cancer Organoid Drug Screen. So what we did in this experiment was to take patient-derived organoids provided by the Hubrecht Institute and perform a drug screen in our organoplate platform. So first I want to introduce you to the organoplate. It combines 3D culture, microfluidic technology, and high throughput. Thus, you can have up to 96 chips per plate and culture multiple cell types. However, in this case, we only have the patient-derived organoid lines. And due to the glass bottom of the organoplate, you can easily do high-quality imaging of your culture. So how do we build our models? We took head and neck cancer patient-derived organoid lines generated in the Hubrecht Institute, and then we suspended them in BME as extracellular matrix. When this BME and cell suspension is liquid, it is added to one of the channels of the microfluidics in every chip. And once it polymerizes, two different compartments are generated in the organoplates. One with the cells and in the gel, and in the other one we will add medium that will be perfused and provide the cells with oxygen and nutrients. So what you can see on the top right corner of the poster is that after 2 days the cells are starting to grow out from the organoids. And then at day 7 of culture, you can already see how the organoids just grew in 3D, and comparing it to the cisplatin treatment, you can clearly observe a difference in morphology between the control and the treated condition. So what I did here is to generate cultures from 6 organoid lines and expose them to 2 different treatments, being cisplatin and targeted therapy niraparib, which is a PARP inhibitor, for 120 hours, and then assess by morphology, cell viability, proliferation, and DNA damage the drug response of each of these lines. So what you can observe on figure A is 2 different graphs, one for cisplatin and the other one for niraparib. The cells were exposed to a series of concentrations, so we built a dose response curve and calculated the area under the curve. And by this, we could generate a little heat map where we have a clear idea of how resistant or sensitive each patient-derived line was. So a quick conclusion from here, or what we can easily see, is that the T6 line, for example, was not responsive at all to cisplatin. However, in the presence of niraparib, the dose response was very, very increased at a very low concentrations, and the line was quite sensitive to this treatment. In figure B, we can see phase contrast images of 3 different organoid lines, and at the bottom, also immunofluorescent staining. What we did here was to recapitulate a potential or mimic patient relapse. And after the cisplatin culture, until day 7, we just exposed the cells to normal culture media, and at day 14, we did the staining for DNA damage and proliferation. What we could see in these 3 different lines is that there is green signal, so DNA damage accumulated in all of the cell lines, but only in the T3 organoid line, there is quite a lot of proliferating cells. This means that after treatment and after some time to relapse, only the T3 cell line is actually still proliferating and could indicate that that patient is more likely to fall into relapse after cisplatin treatment. So what I want to conclude from this poster is that you can create patient-derived cultures from organoid lines and capture interpatient differences in drug response, and potentially use this information from different assays and different measurements to guide therapy selection of at least head and neck cancer organoid lines. Thank you for your attention. If you have curiosity from any other models, just visit mimetas.com, and for any questions, you can contact me in this provided email.